Hello and welcome back to the channel. As always, I am your host, Morgan. This week, we're going to be diving into the topic of my final research project for my herpetology class. So it's something I can kind of ramble on and on about at this point because I've already given a presentation and written a research paper that I'm fairly proud of that I will publish on my website and link down in the description below if you would like to check that out. But I should probably stop teasing you because today our topic is lizard venom. Now, for the first part of our discussion, let's talk a little bit about the order Squamata, which is the taxonomical order in which lizards are found. In this group, we also see the snakes, because snakes are essentially very specialized lizards. This is important because historically, it was thought that snakes were the only venomous grouping in this entire order of reptiles. So as far as venomous lizards went, snakes were the only ones, technically speaking, for a very long time. Now, this did change when in the early 1900s, it was discovered that members of the genus Heloderma were confirmed as venomous. This genus is made up of the Gila monsters and beaded lizards, and their venom has fairly unique properties where it does inflict very intense pain right away, as well as some other symptoms like nausea and some weakening of the area, swelling, and other things that are generally very uncomfortable. And it is thought that their venom is primarily for defensive purposes since they feed primarily upon eggs and other things that do not need to be subdued actively. And if we look over in the snake world, you'll see that in members of genuses that have developed venom previously, the species that stopped preying upon things that need to be actively subdued have very reduced venom or no venom at all anymore. So it is very reasonable to suggest that Gila monsters and beaded lizards venom was specialized for defense. Since if you look at one of these creatures, they're not very specialized to defend themselves beyond wedging themselves in a rock face in their desert habitat. So venom is very helpful. Another clue that it is defensive venom is that very intense pain that has been recorded in several writings to come up almost immediately upon being bitten. Now, the symptoms of Heloderma venom does vary based on the severity of the bite. Gila monsters and beaded lizards have these venom glands in their lower jaws and serrated teeth. So they have to kind of chew the venom into whatever they happen to have bitten. It's not like the snakes that have their fangs on their upper mouths, whether they're rear fanged or front fanged, and a fairly advanced venom delivery system where it's basically a hypodermic needle and pressure that injects the venom. They don't have to chew it in, whereas the Gila monsters do. But we're going to move away from the Gila monsters now and move into the Komodo dragon problem that really has taken over the lizard venom debate for the past few decades. So for a very long time, it was thought that Komodo dragons used a sort of weaponized bacteria, as explained in the bacteria as venom hypothesis, by essentially biting and using the very nasty bacteria found in their oral flora, which is a lot of very big words to describe deadly bacteria living in their spit, to induce some sort of shock and sepsis in large animals to overcome and eventually consume them. This hypothesis was taught for a very long time until some research was done, namely by Fry, where he took scans of a Komodo dragon head, so a preserved specimen, and found a fairly large venom gland in the lower jaw, as well as venom ducts that ran up to the lower teeth. It was fairly similar to the other known venomous lizard species, however, without the serrated teeth and a little bit more advanced. And this got researchers thinking, what if it's not weaponized bacteria? What if it is venom helping lizards like the Komodo dragons and other members of the genus Varanus, so other varanid lizards, other monitor lizards, to subdue prey, especially in these environments where it would be very detrimental to possibly lose an animal or prey item. So like these scrubland and deserts where you might not see a prey item for a very long time, or in these arboreal environments where it's very easy for the prey to get away and you could be in a lot of trouble if it does get away because you need those nutrients to survive. So this kicked off research into looking into these varanid lizards and their venom or lack thereof. And what has been found is that in these lizards, there's a common theme that they do find venom, especially in the arboreal, desert, and scrubland species. And this venom is unlikely to be used as defense because there are no chemicals found that induce pain right away. The venom is, however, similar in that it tends to have these agents that prevent clotting. So one bite and the animal bleeds out. So there has been more work done to try to lay the bacteria as venom hypothesis to rest by other researchers that have taken swabs of captive Komodo dragons at different ages, from hatchlings that have never been fed to older adults, and then compared the bacteria that they found in those swabs. What has been found in these tests is that the bacteria found in the mouth matches the bacteria that was already on the food items offered to the Komodo dragons. And their oral flora was no different than other large carnivores. So any wild Komodo dragons with fairly nasty bacteria is probably incidental, and it's highly unlikely that they are actually using weaponized bacteria. But how is all of this venom kind of related with snakes, helodermatid lizards like the Gila monsters and the beaded lizards and the varanid lizards? Well, Fry did some more work and found in the iguanan lizards like the eastern bearded dragon some venom glands in both the upper and lower jaws. Now, these were not advanced venom glands, and the venom found there was not a sophisticated venom like that of the snakes or the other lizard species. But this finding has proposed this Toxicofera clade that would then lump these groups together in a fairly well-resolved branch of the evolutionary tree, suggesting that venom likely evolved once and then specialized from there, with venom being that basal characteristic or the first characteristic. So in the species that are no longer venomous, that venom was then lost for one reason or another as they evolved because it was no longer needed. Venom is very expensive for the animals to produce. So if it's not needed throughout time, it's likely no longer going to be produced. However, there is still some contention. This hypothesis is still being talked about and tested. There are some scientists that still say that it is unlikely for this particular scenario to have happened and that Venom evolved at least twice in the reptile world, possibly a third time in branded lizards. But there are still some papers being published that are clinging to that bacteria as venom hypothesis and saying that there's no solid evidence for the venom in branded lizards. However, these papers have been noted by more recent studies to be highly biased. So if you do go out and read those, remember that. As of the most recent papers I read, the Toxicofera clade is fairly solid in the herpetology world, and the more evidence they're finding makes that particular hypothesis for the evolution of venom to be more likely. But this is very interesting. This is a field that needs a lot more work. This is still a very new field, and especially with this varanid lizard venom, it's interesting because there's possible medical applications that haven't been explored, especially because they prevent clotting. It's been suggested that it's worth exploring the toxins in their venom for medical applications for stroke and other clotting disease medications. And it's possible that the research into these venoms can fill gaps left by venom research in the snake world. And it's just a very exciting, very new area of research. And as of right now, the best thing that can really be done with this is just to continue on with this research to help tie up those loose ends and learn as much as we can about how venom evolved in the reptile world and the medical applications, of course. But this particular research can help answer some questions about the evolution of the venomous squamates and their close relatives as well. So that's all I really have for you, unless you want me to keep rambling on for an hour or so, but I will cut myself off there. Like I said, I will publish my paper on my website and link that down below for you, as well as link all of my resources in the description down below. Now, a lot of them do come from some academic journals that if you're not logging in with a university ID, you may not be able to read them, but their abstracts at least will be available. So I do hope that you found this interesting. I hope that I wasn't talking you into a completely comatose stupor, and I hope that you have a wonderful rest of your day, and I will see you all in the next one.
Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.